let's begin with inheritance and variation. So, the genetic material in the nucleus of a cell is composed of a chemical called DNA, which I'm sure you're familiar with. DNA is made of a polymer made up of two strands which wrap around each other like a rope. So in cells, we have a nucleus. A nucleus contains chromosomes. Chromosomes contains two strands of DNA like this, and within these strands of DNA we have something called bases. Now, 3 of these bases codes for one gene. So let's write this in another colour. A triplet of bases codes for one gene. What proteins we have. So each gene codes for a particular sequence of amino acids. Okay. Now the genome are all the genes that code for all the proteins. So this is what we're referring to when we say the genome. So now we're going to look at the DNA structure as well as protein synthesis. So the DNA structure. As we said, DNA is made up of two strands which are wrapped together in a structure called the double helix. Now, it stands for deoxyribonucleic acid. And this is a polymer that contains instructions for the body. The DNA is made up of many small parts, and these small parts are called nucleotides. Each nucleotide is made up of one sugar molecule, one phosphate molecule, and one of the 4 types of bases. So DNA is made up of nucleotides, and nucleotides contain one sugar molecule, one phosphate molecule, and 4 bases. The 4 types of bases that we're referring to here are A, T, C, and G, in which A and T always pair, and C and G always pair. Now, each DNA molecule, as we said, is made up of 2 DNA strands which are twisted together. Now, each base is connected to another base along these strands of DNA. So we've got a different base here as a different base, here as a different base, here, etc. They're the different bases. Now, each base is connected to another base in the other strand. So when we mean by A and T and C and G, A will only and C will only pair with G, and then you might have a T and an A, and a G and a C. Now the order of these bases is important. So first of all, this is called complementary base pairing. When A's only react with T's and C's only react with G's. Now the order of the different bases forms a code. So that's why the order is important. Each group of 3 bases codes for 1 amino acid. So let's write that up here. 1 amino acid is coded for 3 bases. Each group of, the amino acids are then joined together to make proteins. So we know that DNA is formed from nucleotides, which are made up of 1 phosphate molecule and one of the 4 bases. We have amino acids. Amino acids are made up from 3 bases. And then finally, we know that proteins are made from the amino acids, because the order of the amino acids and the order of the bases determines what protein there are. Now there are 20 different types of amino acid. Okay, and therefore it is the order of the bases in the DNA. So it's the order of these bases which determine what amino acid it is, and therefore what type it is, and therefore what protein is created. So these are the coding parts of the DNA. This is what we call the coding regions. However, there are also non-coding parts of the DNA that do not code for proteins. Things such as switching genes on and off. They're still important because they still are determining factors in our genes and controlling whether the gene is prevalent or not. Now let's go through the process of protein synthesis. So because the DNA strand is too big to take out of the nucleus and replicate, a copy of it is made, and this is how protein synthesis works. So step 1, you can just say the DNA molecule is too big to move out of the cell, and therefore in step 2, the 2 strands of the DNA separate. So imagine we have this. And now what happens is we have an mRNA nucleotide. Now this mRNA nucleotide then comes to the rescue and forms a complementary strand to one of the other strands. So for example, if one of the 2 strands of DNA that separate look like this, then the mRNA molecule and the mRNA nucleotide will look like this. It will form the complementary sides to it. Then what it does after it's formed the complementary bases on the strand, both mRNA nucleotides that have made copies of either strand are joined together, and they come together to create a template of the original DNA. So we just call it mRNA now. mRNA template for the DNA. So now we have a template for the DNA. We then move on to the next step of moving the mRNA molecule out of the nucleus, and it goes into the cytoplasm and onto the structures called ribosomes. Now, previously we memorized ribosomes as the structure for protein synthesis, so let's see what happens now. So after it reaches the ribosomes, the bases of the mRNA are read in threes. Because remember what we said, 3 bases codes for one gene. 3 bases codes for one gene. So they are now read in threes to code for an amino acid. So remember, the first 3 bases also codes for one amino acid. A triplet of bases codes for one amino acid. And now the corresponding amino acids that are read are then brought to the ribosomes by things we call the carrier molecules. And then these amino acids connect together to form a protein. And then what happens is the protein then has a specific 3D structure as it folds together, and it's unique as well. So we've got the DNA molecule is too big. The 2 strands of DNA separate the mRNA nucleotides, come and make copies of either strand of the DNA. It then joins to form the mRNA. It then leaves the nucleus, goes into the cytoplasm to the ribosomes, where at the ribosomes the bases are read in threes for one amino acid. Then the corresponding amino acids are brought to the ribosomes by the carrier molecules, and then the amino acids then connect together, forming a protein, and then the protein then folds into a unique 3D structure. Okay. Now, there are 3 ways mutations can occur. Changing the bases. So a base can be inserted, a base can be deleted, and it can be substituted. So if a base is inserted, because they're read in threes, every base from this point onwards will change the way it's read. If it's deleted, again the same thing will happen because it will change the amino acids coded all the way after this deletion. And if it's just substituted, it will only affect the bases of this one and this one, not all the way before or after. Now, a change in the type or sequence of amino acids will affect the way it folds. And if it affects the way it folds, it then affects the 3D structure. And if it affects the 3D structure, it affects the active site. And therefore, what it can normally do, its function, is no longer able to do because it's no longer complementary. Okay, so we now understand that variation in organisms occur from 2 reasons. We have the coding regions, which are things like DNA, and then we have the non-coding regions, which are responsible for switching genes on and off. Okay. So let's look at some inherited disorders. So we have polydactyl. Polydactyl is when you have extra fingers or toes. And it just comes about from a dominant allele. Cystic fibrosis is also an inherited disorder. And cystic fibrosis specifically is a disorder of cell membranes which causes mucus issues. Cell membranes which causes thick mucus in the airways and pancreas. And cystic fibrosis is actually caused by a recessive allele. Now, embryonic screening allows scientists to observe whether a child will have genetic defects or not. Now, if the embryo is developed in the lab, then the cells can just be taken, or DNA from the embryos in the womb can be extracted. Now, arguments for and arguments against. Arguments for is that it reduces people's suffering, it allows parents know disorders because treating them might be expensive. Arguments against, it could encourage people to pick characteristics like designer babies. It's expensive to carry out the screening. It could lead to a miscarriage, and decisions about terminating a pregnancy would have been made by this decision, which is quite, baseless and suggesting that disabled people don't have as many rights. Now, sex is controlled by the 23rd pair of chromosomes, because there's 23 chromosomes in total, and the 23rd pair determines the sex. Variation. So genetic variation involves coding regions and non-coding regions, but we also have other types of variation. So variation in terms of the phenotype, which are the physical characteristics of an organism, sprouts because of two things. One is the genotype. Now, the genotype is the genes inherited. So firstly, this looks at genes inherited from the parents, the combinations of the genes, and the fact that the only people that have the same genotype would be twins or triplets, etc. Now, the other reason for phenotype variation is the environment, which is the place it lives in. So the conditions in which that organism lives and grows can affect its phenotype. And also, for example, let's look at scars in animals, or being tanned, or your hair color changing due to the sun, things like that. Now, sometimes characteristics can result from a combination of genetics and the environment, such as weight, which depends on how much food you eat, but also how quickly your body breaks it down. Now, genetic variation is introduced by mutations in the sequences of DNA. So why do we have genetic variation? Let's look at it now. So, genetic variations happen due to mutations that occur. Now, mutations then mean that every time the genotype is slightly altering. However, most of the time, this does not have effect on the person. Now, if the mutation determines the phenotype and is advantageous, the selection will mean that it will become a more common phenotype. Okay. Evolution is a change in the inherited characteristics of over time through a process called natural selection. Okay. Let's look at evolution. So evolution, in terms of the definition, evolution is the change in characteristics that are inherited by a population through a process called natural selection. So evolution rides on this idea that we change our characteristics through a process called natural selection. Now, the theory of evolution states that all species have evolved from a single life form. Billions of years ago. Now, evolution happens because of natural selection. So, in terms of the process of evolution through natural selection, what the theory is that a mutation occurs, a random mutation occurs due to variation that we looked at, the constant swapping of genes, and this mutation, if it provides a survival advantage, then that means the organism is more likely to survive. Now, if the organism is more likely to survive, then it will breed, and the mutation will be passed on. Over many generations, the frequency of this mutation will only increase, and the gene will fully develop. Now, this may cause one population of a species to become so different that they no longer even look or are. They're no longer able to produce fertile offspring with what they once were. And this is called speciation. It's how a singular species, due to a mutation or variation because of, physical separation, it then becomes so different because one gains survival advantages that both of the new species created cannot reproduce with the old species that it once was, and therefore that's how you know that a new species has been created. Now, we also have selective breeding. Now, selective breeding is when humans choose which organisms to breed in order to gain desired characteristics. So, this is the structure. So, first of all, the parents with the desired characteristics are chosen. They are then bred together. Now, during this stage, there could be animal discomfort because it's unnaturally forced. And then they then have produced offspring. And the offspring, only the offspring with the relevant gene will then be rebred. And this will happen over many generations until this is fully developed. Another issue with this, however, is that it could lead to inbreeding, because if you're only having the relevant gene breed with the relevant gene, then this can lead to breeding within too close of blood, and therefore inbreeding can cause some issues, because breeding those with similar desired characteristics mean you're reducing the gene pool, as a number of different alleles reduce. So if the environment changes or if there's a new disease, the species could become distinct if they all have a susceptibility towards it. Extinct. And also another issue of inbreeding and reducing the gene pool is that there's an increased chance of genetic defects, such as the mad cow disease. Now, what is genetic engineering? So selective breeding is choosing characteristics and breeding them together. Now, genetic engineering is physically modifying the genome of an organism by introducing a gene of another organism to just modifying the genome with another gene from another organism to create a desirable characteristic that perhaps can't be bred. So the process of genetic engineering. Firstly, the genes from the chromosomes. Let me write this in smaller font. So firstly, the genes from the chromosomes that we have in the main cell are cut out. And then we have a plasmid, or sometimes a virus, is used to then is cut via the same enzyme. So we've got the gene, which is cut from the enzyme, and then the plasmid, which is cut from the enzyme. And then what happens is that we have sticky ends on both of them. So imagine, because of cutting it, we now have sticky ends. So what happens is the loop and the gene, plasmid is a DNA loop, so the plasmid and the gene have sticky ends, and they then join together. The sticky ends join together by DNA ligase enzymes, so just by DNA enzymes. Now the combined loop is placed in a vector. So this vector could be a bacterial cell, etc. And in this bacterial cell, they will then multiply as the intended gene, as the modified gene. The colonies that will be there will be the bacteria that are also producing the modified gene as they were inserted together. Now, in plants, the vector is what we call meristematic cells. So in plants, the vector used are meristematic cells, which are cells in the meristem, so stem cells basically, which then produce copies of the identified plant. So let's just go through them again. So we have the gene. Imagine we have the gene here, and we have the chromosome here. Now the gene is then cut out, and the chromosome is cut out. The gene is cut out, and the plasmid is cut out using the same enzyme. They are then put together using a DNA enzyme. The sticky ends created are put together, and then they are then placed within a loop. So they're then placed within a vector. This vector can be bacterial cells, or if it's a plant, then it's meristematic cells. And then this combined loop here then reproduces and reproduces, and then as the cell reproduces, this gene becomes more varied, more popular. And that's genetic engineering. Now, genetically modified crops are what we call GM crops. Genetically modified crops are engineered to be resistant to things like herbicides, insects, and they will result in increased yields, therefore increased profits. Now, it can also be used in medicine to cure inherited disorders. It may be possible to use genetic engineering to cure things like being paralyzed. Now, it's called gene therapy. It may be possible to use genetic engineering. Okay, so the perceived benefits is that it can be useful in medicine to mass produce certain hormones in microorganisms. However, GM crops might have an effect on wildflowers and insects because they are infertile and these genes could spread into the wild plants, leading to infertility in other species, which could affect the entire environment. And growing with herbicides and pesticides can kill insects. In agriculture, though, it can be improved to improve growth rates, introduce modifications that allow crops to grow in different conditions, and introduce modifications that allow plants to make their own pesticides and herbicides. Crops with extra vitamins can be produced in areas which are difficult to obtain, such as the golden rice, and greater yields can help solve world hunger. However, some disadvantages is people are worried that we do not completely understand the effects of GM crops, and genetic engineering in agriculture could lead to genetic engineering in humans, which is dangerous. Finally, cloning. So, the different types of cloning. Cloning is just creating genetically identical things. So we have firstly tissue culture. So tissue culture is used to preserve rare plants. So what happens is plant cells are taken, they're then placed in a growth medium. So placed in, for example, a nutrient broth with lots of nutrients available to new plants. And they are identical to the parent plant. So that's what tissue culture is. You just take the plant cells and put it in a nutrient broth, and then it will be genetically identical, but it's used for preserving rare plants. And it will then produce a plant that's genetically identical. Now, cloning in animals is also prevalent. So there's adult cell cloning, and on top of adult cell cloning, we also have embryo transplants. So, embryo transplants is when a sperm cell with desirable characteristics and an egg cell with desirable characteristics are taken. They then fuse together, create an embryo. And this embryo here then divides many times and is then inserted into the host mother, also what we call the surrogate. So surrogacy is what we call embryo transplants. When you take a sperm cell, you take an egg cell, you create an embryo, you put it in a host mother, and then the offspring produced is genetically identical with a desirable feature. That's embryo transplants. Adult cell cloning is another thing. So adult cell cloning is when you remove the and you remove the and then you remove adult you move the adult body cell shell, and then you then put the adult body cell shell around the nucleus, you're then literally creating a cell. And the cell then begins to divide and form an embryo. So you take the nucleus of an egg cell, you place it in an empty adult body cell, add an electric shock, and boom, it starts to divide. The embryo is then implanted, and the offspring is actually a clone to whoever's nucleus that once was. Now we discussed the theory of evolution a bit more. Okay, we're almost done. So the theory of evolution is that variation exists within a species because of the fact that we've all evolved from the same life form and things like that. Now, variation exists due to mutation. Organisms that are most suitable to the environment survive. So that's this idea of survival of the fittest. However, there was controversy to his ideas because it contradicted the idea that God was the creator and there was not enough evidence for him. Other opinions, such as Jean-Baptiste Lamarck, thought that changes that occur during the lifetime were passed on to an offspring. So if an organism repeated an action, then eventually that characteristic would develop further. However, it was wrong because we then understood that it was not passed on. Developments in the environment were not passed on in sex cells. We talked about speciation, the process of new species forming through the natural selection of different alleles, and this increases the genetic variation until the new population cannot breed and they are therefore infertile, and that confirms that it's a new species. So in terms of the exact process of speciation, we've got variation exists. Let's do it here. So variation exists within a population as a result of variation exists as a result of genetic mutation. And the alleles which provide the survival advantage are therefore selected through natural selection, which we looked at the processes. So variation, the alleles providing the best survival advantage are selected, and then the population of a species can become isolated. So the population becomes isolated, could be by physical obstruction or something together. Now the alleles may be advantageous in the environment, so they may require different alleles. Over time, the selection of the different alleles will increase the genetic variation between the two populations. So over time, there'll be increased genetic variation between the two populations. And therefore, when they're no longer able to breed together, a new species is formed. Now, Gregor Mendel carried out breeding experiments on pea plants using smooth peas, wrinkled peas, green peas, and yellow peas, and observed how the offspring see which characteristics are inherited. He then kept a record and viewed the characteristics as hereditary units, which we call genes. In the late 19th century, the chromosome, as part of cell division, was observed. In the 20th century, it was understood that chromosomes and units had similar behavior. So evidence for evolution is seen through 1. Fossils, and 2. antibiotic resistance. So fossils are formed when the plants of organisms have not decayed because of the oxygen or moisture that was not present. So there's a dry conditions and the microbes could not decay. The microbes that could cause decay could not survive, and then parts of an organism such as teeth, shells, and bones were replaced by minerals as they decay, forming a rock structure. And the preserved traces, such as footprints, burrows, and rootlet traces, remain due to ground hardening. So fossils are produced when the microorganisms to break down are not there. So there's a lack of oxygen. It's a cold environment. So therefore, any bones just turn into minerals and preserved in rocks, and then your footsteps and traces like that are kept due to ground hardening. Fossils then show the anatomy of organisms and how it's changed over time, and they can be used to compare how closely related two organisms are through looking at the number of similarities. Fossils cannot be used to tell us how life started because most early life forms were soft bodied, and also any traces of them have been destroyed by geological activity. Extinction is when an entire species has died out because of changes in environment, catastrophic event, destruction of a habitat, new predators, new diseases competing with the species. Resistant bacteria. So bacteria produce at a fast rate. Now the mutations that saves you from something, you are going to die. So exposure to the antibiotics creates a selection pressure, as those with antibiotic resistant genes and those without will die. As a result, those with the antibiotic resistant can then reproduce and pass on this gene, and this population only increases, and the bacterial diseases spreads rapidly. For example, MRSA, called the superbug. Now, to slow it, we can not give antibiotics for viral diseases, finish the courses of the antibiotics. They should be used less in agriculture, and specific ones should be given to specific bacteria. Now, classifications of living organisms. I know we're going over a little bit, but we're almost done. So, we have the Linnaean system, which Carl Linnaeus put forward in the 1700s. And this just suggested that living kingdoms were split up into different groups. So, what kingdom, phylum, class. Now, this was a Linnaean system. The binomial system gives each organism a name, which comes from its genus and then its species. The three domain cyst groups above kingdoms. So above kingdoms, Carl Wolsey added three things: archaea, bacteria, and eukaryota. He said that all kingdoms would form in one of the three. So archaea were the primitive bacteria, so the extremophiles. The bacteria are just a true bacteria, which we see today. And the eukaryota are organisms who have a nucleus enclosed in the membranes. And that is topic 6 done. And finally, we're moving on to topic 7, which is ecology. So ecology. Okay. So this topic is a little bit less dense than the previous one. So an individual is part of a species, but lives in a habitat within a population. So what we have here is everyone lives in different populations. These things are called populations. Now within the population, we have small habitats. And then different species live in different habitats. So when different populations... Oh, sorry, it's the wrong way. Okay, 1 second. So an organism would live in what we would call a population. And this population will be within a habitat. Now the different populations will interact with each other. And this then creates a community. An ecosystem is the interaction of a community with non-living parts of the environment. So let's see what that means. So you've got an ecosystem. An ecosystem is the interaction of a community with abiotic components. What that's then saying is it's basically how a community, so a group of different populations within a habitat, interact with the conditions of their environment. So organisms which need the same resources, therefore, light, water, mineral ions, food, mating partners. So interdependence is another term that's important. Interdependence is when all of the components within an ecosystem depend on each other. So organisms depending on one another. Depending on one another. Now, interdependence is used for things like food, shelter, reproduction, pollination, and seed dispersal. So the removal or addition of a species to the community can affect the populations greatly. So a stable community, remember communities are how the different populations communicate. So a stable community is one where all of the biotic components and all of the abiotic components are in balance. So biotic components are when the not biotic components. A stable community is when the biotic components are in balance with the abiotic components. Therefore, when the population sizes remain constant and when they are lost, it's very difficult to replace them. So let's look at some common abiotic factors. So abiotic factors. So the first thing we see in terms of abiotic factors is light. Plants compete for light. Plants compete for, in terms of temperature. Temperature affects plants. We can look at soil pH, moisture levels, as well as soil pH, the nutrient level within the soil, wind direction and strength, carbon dioxide levels, oxygen levels for aquatic organisms. So these are loads of abiotic factors: light, temperature, soil pH, moisture levels, wind, carbon dioxide, oxygen levels. Okay. So these factors can all affect a community. Now, in terms of biotic factors, living factors, we have things like food availability, new predators, new pathogens as well, competition. Now let's look at some adaptations that we have. Because organisms have adaptations which help them to survive and are suited for their environment. So there are things such as structural adaptations, there's behavioural adaptations, and then there's functional adaptations. So in terms of structural, we have things such as the shape, being able to camouflage, how some animals have a thick fat insulation layer. That's all comes under structural. Behavioural, on the other hand, is things like how an organism behaves, such as pretending to be dead to avoid predators. Courting behaviour to attract male or female. Now, functional adaptations are things such as processes such as reproduction and metabolism. So conserving water, conserving heat, conserving late implantation of embryos, and things like that. Okay. So we're now going to look at levels of organization. So in terms of levels of organization, it always begins with the producer. Now, the producer is the normally plant life that photosynthesizes. And they're always the photosynthetic organisms, that's what we like to call them. So we've got the producer at the bottom. We then have primary consumers. The primary consumers obviously eat the producers, so energy is transferred in the organism when one is eaten by another. And then what eats the primary consumers? The secondary consumers. The secondary consumers would then be viewed as carnivores because they're eating meat. So we've got the primary consumers, and then we've got the secondary consumers, and then we've got at the top the tertiary consumers, which tend to be the apex predators, the things at the top of the food chain. Now, to work out the distribution in abundance, you can use transects and quadrats. A stable community will show population cycles between predator and prey. Now, we also have material water. So we've got condensation in water, precipitation in lakes, seas, and evaporation from the seas and lakes as well. Things that can affect the rates of decomposition include the temperature, the moisture levels, so temperature, chemical reactions, because they are enzyme-controlled reactions when you're decomposing via microorganisms. Therefore, you do not want it to denature and stop. Water, microorganisms grow faster in conditions with water because water is needed for respiration. That's why microorganisms do better with moisture. And finally, oxygen levels, because again, through respiration, aerobically. Now, decomposition can be used in things such as compost. So compost is when basically the production of biological matter decaying. So anything biological in its decaying form is what we call compost. It's used by gardeners and farmers. It's a form of natural fertilizer. It's actually very good. To do this, they have the optimum conditions for decay. So to enhance this, you need to ensure that oxygen levels are reached, and the increased temperature increases the rate of decay, so increasing the temperature as well. Methane gas. Microorganisms decompose waste anaerobically. So when there's not enough oxygen, then it has to go through the anaerobic route. Methane gas, which can be burnt as a fuel, so hydrocarbon. Now, biogas generators are used to produce methane. They require constant temperature and cannot be stored as a liquid. You can investigate the decay on milk using that required practical. Biodiversity is a variety of organisms or species within Earth. So there's quite a lot. Now, the future of Earth depends on maintaining biodiversity. So deforestation, pollution, sewage, smoke, landfill, we have to keep this in mind. Now, in discussing compost, it's also important to discuss peat bogs. So in terms of peat bogs, peat is a material formed when plant material has not fully decayed, as there is not enough oxygen. So peat is actually a very bad thing. So peat is not fully decayed plant material. And the reason why peat is not fully decayed is because of a lack of oxygen. Now what happens is this then accumulates in the bogs, and because of anaerobic respiration it then releases acid, just how we release acid. So then the conditions become acidic as well as waterlogged. Because there's not enough oxygen, it can therefore not respire as well, and therefore there's no point using the water. These bogs are a habitat for many species, so it's then used as a habitat for many species, especially extremophiles. However, we tend to remove these peat bogs in order to get more peat as compost or dried fuel. And however, when they are destroyed, they produce so much carbon dioxide into the atmosphere, it's crazy. Deforestation, the slash and burn technique. Global warming. Interactions with the ecosystems such as maintaining rainforests, reducing water pollution, preserving scientific areas of interest, replanting. Negative impacts, so production of greenhouse gases, production of sulfur dioxide, which causes acid rain. So sulfur dioxide is responsible for acid rain. Recycling rather than dumping waste in landfill reduces the amount of land taken up. And finally, we have trophic levels. So level 1 are organisms at the first level, such as producers, and then level 2 would then eat the next one. Now, pyramids of biomass show the relative biomass at each trophic level. It shows the relative weights. So there's less biomass as you move up the trophic levels because not all the food is converted. This means the biomass in the level above will always be higher because only approximately 10% of the biomass eaten is converted to the next trophic level. Factors affecting food security, things like increasing birth rate means that more food is required, changing diets, new pests and pathogens, climate change, conflicts. Farmers aim to increase the amount of energy that is converted to biomass in livestock because this is more inefficient. So farming techniques, things like raising animals in small cages so there's less movement and less energy is wasted. However, this is very bad for animals and has many ethical objections. Sustainable fisheries. This is because humans are fishing at a faster rate, the number of fish are decreasing. So to avoid this, you have things like net sizes, fishing quotas. Biotechnology. Biotechnology can be used to help feed the population and potentially provide treatments. So let's look at the fungus mycoprotein. The fungus fusarium is used to create mycoprotein, which is protein rich, suitable for vegetarians, grown on glucose syrup in aerobic conditions, is harvested and purified so it can be consumed, and it's protein without having to eat the animals, which reduces. This source of protein is good because there's no need for animals, so we can reduce the amount of livestock because the amount of cows in the world just used for cattle are very bad. Also, we can genetically modify bacteria, for example, insulin taken to treat people with diabetes. Genetically modifying crops to have certain properties that can have many advantages. For example, modifying them to increase their nutritional value. And that is this topic complete. We now move to organic chemistry. So yesterday hydrocarbons are purely hydrogen and carbon. Now, it's a compound that's formed by carbon and hydrogen only. So we have different types. We have the alkanes. Now, alkanes are examples of homologous series. Homologous series are things that have a similar structure and therefore react quite similar way. So alkanes have the structure CnH2n plus N. We have the structure monkeys eat peanut butter, methane, ethane, propane, butane. And it'll be like CH4, C2, H6, C3, H8, C4, H10, and things like that. Now, hydrocarbon properties change. So let's try and see if we can remember the properties. So, shorter chain hydrocarbons, in terms of, we need to look at the properties in terms of flammability. We need to look at its consistency, how runny it is, basically. And then we need to look at its volatility, which is basically its melting point. So we know that short-chain hydrocarbons are more flammable, are more runny, and have lower melting points. Yep, so that's correct. Consistency of more runny, that's correct. So they're less viscous, and the more flammable is, that's correct. Now, hydrocarbons can undergo combustion. So hydrocarbon plus oxygen would burn and create carbon dioxide as well as water. However, this is if the conditions are provided which will enable complete combustion. Or complete combustion. And some carbon monoxide can be produced instead. So fractional distillation, that's where we got up to yesterday. So let's draw the diagram. So in terms of fractional distillation, we have the fractionating column, so what enters, what exits at the bottom here. And what enters as a liquid is the crude oil. This is what we call crude oil. So the crude oil enters this way. Now it goes highest boiling point, lowest boiling point. Yep, so it's hottest to coolest. And so let's create that effect here. And how it's hottest to coolest. So now, this is a fractionating column. So what we tend to notice about the fractionating column is that liquids with similar boiling points tend to stick together. But because we know that depending on the length of the hydrocarbon, the properties remain similar, so we'll note that the ones with the lower boiling point are going to be shorter chain hydrocarbons. So we'll also be in order of short chain hydrocarbons to long chain hydrocarbons. So the crude oil is heated, most of it is turned into gas, and there's a temperature gradient. However, some of it is not turned into gas because it's got a very, very high boiling point, and that liquid is what we call bitumen. Bitumen has a very high boiling point, and therefore it is not even turned into a gas. The rest enter the fractionating column, and in the column there's a temperature gradient. So The longer the hydrocarbons, the higher the boiling points there are, and then the shorter ones have a lower boiling point. So we're going to need to know roughly which one goes where. So the ones which remain a gas go here, go this direction. And the ones which all condense into liquids go here, 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 and here. Now these are all specific liquids. So the one that remains a gas is what we call liquefied petroleum. Then on each of these ones, where we have each of these fractions, we see petrol in the first one. Then we see kerosene, which is aeroplane fuel. Then we see diesel. And then we see heavy fuel oil. So if we go to that again, the ones that remain a gas are liquid. The ones that remain a liquid are bitumen. The one that stays as a gas because it's a very low boiling point, liquefied petroleum gas. Then we have petrol, kerosene, diesel, and then heavy fuel oil. Now, crude oil has various uses. It can be used for loads of things. The petrochemical industry, which uses hydrocarbons for feedstock to make new compounds. All the products you get are organic compounds. Now, so when we're talking about organic chemistry and organic compounds, organic just means anything that comes from carbon. And that's what we mean. Okay. Now we have a method called cracking. So cracking is a way in which we can convert long hydrocarbons into short hydrocarbons, because long chain hydrocarbons are not very useful with its properties. They're not very good fuels. So if you wanted good fuels, you can undergo a process called cracking where this will be possible. So we can see here that the properties, just as a reminder, short chain hydrocarbons are flammable, which is really good for being fuel. They have a low melting point, which is good because you don't have to heat them to a very high temperature for them to do it. And they are more runny. And then the long chain hydrocarbons have opposite ones to that. Now, what happens when we crack something is that we not only produce the short chain hydrocarbon that we want, we produce something else. So when we crack an alkane, we not only produce a shorter chain alkane, but we also produce something called an alkene. And this introduces us to the next molecule. So just a bit of further information. So cracking is therefore a thermal decomposition reaction. The reason why is because energy is needed to break the bonds between alkane to produce a smaller alkane and an alkene. Now, alkenes have the chemical structure CnH2n. Alkenes have CnH2n plus 2. And then it just works that way. So, away in terms of steam. So an alternate method of cracking is vaporizing them and mixing them with steam. Okay. So vaporizing them and mixing them with steam. And this is called steam cracking. So there's catalytic cracking, which is the one we just went through, which is using a catalyst and so what happens is, the method of this cracking, we do need to know the method of this, is that the long chain hydrocarbon is heated and vaporized, and then the vapor is passed over the catalyst, which is aluminium oxide, and then the longer chain molecules then split apart on the surface of the catalyst, producing the shorter chain ones. Now let's look at alkenes. So we've looked at the formation of alkenes and where alkenes come from. So alkenes come from when they've been cracked. Now we've looked at the difference. So alkanes have the structure CnH2n plus 2. So it will look like this. However, alkenes, so these are saturated molecules, whereas alkanes are not saturated. The reason why they're not saturated or unsaturated is because they form CnHC, so for methane, H, 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 H. But we know that carbon has 4 bonds, so we therefore have a double bond here, and therefore it's unsaturated. Because here we're only using 1 and 2 and 3, and for this carbon atom we're only using 1 and 2 and 3, so we add another bond here because there's a spare bond. Now the first one here is not actually methane, not actually called methane, it's just called ethene. So ethene, propene, butene, pentene. Now in large amounts of oxygen, alkene can combust completely. So alkene can combust completely just to produce water and carbon dioxide. Similarly, just to how alkanes can produce water and carbon dioxide as well. However, when there isn't enough oxygen, then there can be incomplete combustion, and this is only with alkenes. So incomplete combustion can lead to carbon dioxide, water, and also carbon and carbon monoxide. So when we have alkene and oxygen, and there's incomplete combustion, you do get water and you do get carbon dioxide, but you also get carbon and you get carbon monoxide. And these two are unnecessary. They also result in a smoky yellow flame. And less energy is being released as well as a fuel. It's not as useful. Okay, so that's when we know the difference between combustion and the saturated and unsaturated. Now we're going to just look at reactions of alkenes, addition polymers, alcohols, carboxylic acids, and condensation polymers. just want to start afresh. So I'm just going to get rid of this. Okay. So we're now going to look at reactions of alkenes. So alkenes can react with a lot of different things. Okay. So reactions of alkenes. So the first reaction that we need to know are addition reactions. So addition reactions, before we get started into addition reactions, let me just tell you what a functional group is. So a functional group is a group of atoms within a molecule that determines how a molecule will react. So any homologous series, which is a group of compounds that react a similar way, will have a functional group that will determine how it will react. Which, for example, alkane's functional group is this. Alkene's functional group is this. So let's look at the additional reaction. So all alkenes have the same functional group, which is this. That's the functional group for alkenes. So they will all react in similar ways. So firstly, we need to know how alkenes react in general. Most of the time, alkenes react via addition reactions. So the carbon-carbon bond, for example, we have this. It will then form C. The carbon will open up its bond and form H, or in this case that'll be Y, H, X, R, H. It'll form like that. So the carbon-carbon bond will double and open up. So that's just the basic addition reaction. Now we have specific addition reactions, so that are quite important to know of. Now, specific addition reactions include things like addition reaction of hydrogen, so hydrogenation. So this is hydrogen, and this is then double bonded, and the double bond opens up. So let's have the same structure. And we're reacting this with hydrogen. We then open the double bond, and it will just produce that. And a lot of these are catalyst reactions. So hydrogenation is just reacting it with hydrogen, and it will look like that. Then we have another reaction which is when we react alkenes with steam. And alkenes with steam produce alcohol. Let's see this in action. So when alkenes react with steam, water is added across the double bond. So you're basically doing the alkene plus water. And when you're doing that, you're going to get this form. The carbon needs to have 4 bonds, and this is the functional group for alcohols. So already looking at this, in order to get to the position of alcohols, you first need to crack alkanes, and then you need to react the alkenes which you have with water, and you then get alcohols. Now alcohols can also react, I think, to get carboxylic acid. So ethanol and water have a higher boiling point than ethene, so both condense, while any unreacted ethene is recycled back. So the alcohol can then be purified. And then halogens react with alkenes as well. Let's just go down. Halogens react with alkenes. Now, alkenes also react in addition reactions with halogens, such as bromine, chlorine, and iodine. So, for example, this structure plus bromine would then create diobromoethene, because this is ethene, bromine. So what you say is you call it diobromoethene. Now, bromine is actually quite interesting when we're talking about alkenes, especially when we resonate it with the colour orange. So let's look at how it's related to the colour orange. The addition of bromine to a double bond can actually be tested for alkenes. So bromine is used as a test for alkenes, to test whether they're alkenes or alcohols. So, how does this work? Well, first we have orange bromine water. And what you're going to do is you're going to add this to a saturated compound. For example, an alkane, basically a compound that is unable to change its structure. Now, if you add it to a saturated compound, then obviously no reaction will happen because it's saturated. However, if we react it to an alkene, we know from the reaction above that alkenes do react with bromine and then create a colour change. So when we add an alkene, it will add across the double bond and then form a colourless solution. So you know that with bromine water, if we add an alkene to it and it turns colourless, then we know it's definitely an alkene. Now let's look a little bit more at addition polymers. polymers are long molecules formed when lots of small molecules join together. These small things are called monomers. So when loads of monomers join together, we call that a polymer. Plastics are made up of polymers, which are usually carbon-based. So the monomers that make up addition polymers usually have a double covalent bond. Therefore, we're saying that they're alkenes. Lots of unsaturated monomer molecules, alkenes, can open up their double bonds and join together. So, for example, if we had many single ethenes like this, then eventually it will form this. So many single ethenes, it will then open up its bracket and look like that. And then it will still have hydrogen, 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 and then it'll be to the power of n. So this is called a repeat unit. So to get from the displayed formula to the repeat unit, you have to draw out the repeating bit and get rid of the two bonds through the brackets and put them through the carbons. Poly. Now we're going to look at alcohols. So alcohols have the OH function. And we know that alcohols are formed when alkenes react with steam and produce alcohols. Now alcohols therefore have the OH ending. Remember the C to C to O to H. That's what the structure looks like. So if we have methanol, for example, by the way, alcohols have the functional group CN H5OH. That's the functional group for alcohols. So if we were to do methanol, it would be C and then H5, so H, 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 O, H, H, H. That's what methanol would look like, and etc. So instead of so the first four alcohols have similar properties. We know that alcohols are flammable. And they undergo complete combustion in the air to produce carbon dioxide as water. So it's important to note that all hydrocarbons that undergo combustion seem to produce carbon dioxide and water if it's complete. If it's not complete, then the alkenes will produce carbon and carbon monoxide. So all of these solutions, methane, propene, methanol, ethanol, propanol, and butanol, are all soluble in water. And therefore are neutral. Now, alcohols can be oxidized by reacting with oxygen to produce a carboxylic acid. So different alcohols form different carboxylic acids. So if you react an alcohol with oxygen, you can then get a carboxylic acid. Now, this carboxylic acid can then be used for many things. But first, before we move on to that, we can also talk about ethanol. So the alcohol ethanol is used and is made by fermentation. So it's found in alcoholic drinks. So fermentation is when you have sugar, and through the help of yeast, the sugar turns into ethanol. The sugar, being the glucose, turns into ethanol and carbon dioxide. Now, fermentation happens fastest at a temperature of 37 degrees, slightly acidic and under anaerobic conditions, no oxygen. So this is how plants anaerobically respire. Now let's look at carboxylic acids finally. So carboxylic acids are a homologous series which have the Cu function. So just going back over this, we have the functional group, and then we have the structure. So let me write alkanes. So alkanes has a functional group CnH2n plus 2, and just a single bond like that. Then we have alkenes. Alkenes are formed by cracking alkanes, and then you get CnH2n, and that's the double bond here. Then you get alcohols. Alcohols are formed when you react alkenes with steam, and you then get CnH5OH, or the structure COH, or the OH, more like the OH function. And then you get carboxylic acids, which is when alcohols react how do alcohols react? Carboxylic acids are produced from alcohols, but what else are they produced by? Let's see. Oh, oxygen. So alcohols just react with oxygen to produce the carboxylic acids. Carboxylic acids have a formula, but we don't need to necessarily know the formula. Okay, so we oxidize it. We don't know the necessary carboxylic acid function, but we know that it will look like this, the functional group. Now, carboxylic acids can also react with carbonates. So carboxylic acids can react with carbonates, just like any other acids, to form salt, water, and carbon dioxide. So if you were to do ethanoic acid is a carboxylic acid. If you're reacting this with sodium carbonate, for example, you're going to get sodium ethanoate plus carbon dioxide plus water. However, we've got one more chemical created. So carboxylic acids dissolve in water. When they dissolve, they ionize it and release H plus ions, resulting in an acidic solution. However, they don't ionize completely. They just form weak acidic solutions. So carboxylic acids do not fully ionize. Now, carboxylic acids can also give rise to esters. Now, esters have a functional group of COO, Cu, and it's formed when a carboxylic acid reacts with an alcohol. It then produces an ester and also water. Finally, we have the harder ones. So these are all the addition reactions. They're the easier ones. We have condensation polymers. Condensation polymers involves monomers which contain different functional groups. So for each new bond that forms, a small molecule is lost. That's why it's called condensation polymers. So what happens is the simplest type of condensation has two different types. So you have a polyester can be made from condensation. This is quite difficult, so I recommend learning this one yourself. So the difference is that with addition, there's only one monomer, which contains a double carbon-carbon bond, whereas condensation can be two monomer types containing two of the same, or one monomer type with two different functional groups. Naturally occurring polymers such as amino acids, proteins, DNA molecules, and simple sugars. Okay, we're now going to move on to the next topic, which is chemical analysis. We're quite good with chromatography, then tests for gases, anions, cations, flame emission spectroscopy, then chemistry of the atmosphere. So let's just do some chemical analysis revision. So, purity is defined differently in chemistry as to every other thing. So when you refer to a substance as being pure, you mean that nothing has been added to it. It's in its natural state. Now, in chemistry, a pure substance that only contains one compound or element. A chemically pure substance will melt or boil at a specific temperature. So, in chemistry, we have pure substances, which is just the substance and nothing else. Now, we know it's pure because it will boil or melt at a specific point every time. Now you can test the purity, therefore, of a sample by measuring its melting and boiling point and comparing it with the melting and boiling point of the pure substance. The closer your measured value, the purer your sample is. Impurities will lower the melting point and increase the range. Impurities can also increase the boiling point and may result in your sample boiling at a range of temperatures. Formulations. What are formulations? We've looked at what a pure substance is. What is a formulation? Well, formulations are useful mixtures. They have a very precise purpose that are made by following a formula. So each component in a formulation is present in a measured quantity. So formulations are basically compounds created very specifically, so there's specific measurements and specific quantities and things like that, all for a intended purpose. For example, let's look at some examples of formulations. So if we look at pigments, pigments give the paint colour, and it's used for purposely bring out a specific paint colour. We have solvents. Solvents are used to dissolve the other compounds and alter the viscosity. Additives. Additives are added to further change the physical and chemical properties. Okay. Now, depending on the purpose of the paint, the chemicals used and their amounts will be changed so that the paint produced is right for the job. In everyday life, formulations can be found in cleaning products, fuels, cosmetics, fertilizers, metal alloys, and even food and drink. So when you buy a product, you might find that it has information about its composition, on the packaging. For example, the ratio or percentage of each component. This tells you the product's formulation. Now we're going to look at tests for gases and anions, and then tests for cations, and then the final few topics. Okay. So tests for gases and anions. There are 4 tests for the 4 common gases, and then the test for anions, and then the test for cations. So you're going to need to know all of them. I'm just going to reload this page because it's being a bit awkward. Okay. Apologies for the internet delays. Okay, so we were looking at the different tests for the gases. So tests for gases. As we said, there are 4 tests for gases. So the first test is the chlorine test. Chlorine bleaches damp litmus paper, turning it white. It may turn red for a moment first. That's because the solution of the chlorine is acidic. So the test for chlorine is the litmus paper test. So we'll go through the tests again. So chlorine is a litmus paper test, turning it white. We've got oxygen. Oxygen is the glowing splint. So if you put a glowing splint inside of a test tube that contains oxygen, the oxygen will relight the glowing splint. Then we have carbon dioxide. The test for carbon dioxide is bubbling it through an aqueous solution of calcium hydroxide, known as lime water, and it will cause the lime water to turn cloudy if it truly is the carbon dioxide. And then finally, the hydrogen test. The hydrogen test is if you hold a lit splint at the end of a test tube containing hydrogen, and you'll hear a squeaky pop, confirming the presence of hydrogen. So just going over those one more time. The test for chlorine involves using damp litmus paper, and it should bleach damp litmus paper white. The test for oxygen is putting a dimly lit splint into a test tube of oxygen, and if it relights, then it shows that oxygen is present in there. The test for carbon dioxide is bubbling bubbles of carbon dioxide through calcium hydroxide. Calcium hydroxide, which is also known as lime water, and it should turn cloudy, so precipitate should be formed. And then the hydrogen test is you have a test tube of hydrogen, and if you put a glowing splint, then it should create the sound of a squeaky pop. So that's the test for gases. Now look at the tests for anions. So dilute acid can help detect carbonates. So carbonates are substances that contain the ions CO3 and negative -2 charge. So these are the ions. Carbonates are anything with this ion. You can test whether a mystery solution contains carbonate ions by putting a sample within a test tube and then using a dropping pipette to add a couple of drops of dilute acid. You should then connect the test tube to a tube of lime water. If the carbonate ions are present, carbon dioxide will be released and will turn the lime water cloudy. So another one for lime water is carbonates. So we know that if we react carbonates with dilute acid, then if it truly is carbonate, carbon dioxide will be formed, and that can be used to turn lime water cloudy. So that's how to test for carbonates. In order to test for sulfates, you can use barium chloride. So with barium chloride, you want to identify the ions SO4 2-, and you're going to do this by adding dilute hydrochloric acid. And again, and then add also, so not only dilute hydrochloric acid, but a couple of drops of barium chloride. Now the reasoning behind this is that if sulfate ions are present, a white precipitate of barium chloride will form. So if the sulfate ions react with the dilute hydrochloric acid, then these sulfate ions will be present, and then barium chloride will form a white precipitate. Now to test for halides, in order to test for halide ions, you just need to add a couple of drops of dilute nitric acid, followed by a couple of drops of silver nitrate solution. Now, the end goal here is that, for example, you have silver and chlorine, then you'll get silver chloride, and this will give a white precipitate. If you have silver and bromine, you get silver bromide, and this gives a cream precipitate. And if you react silver and iodide, then you get silver iodine, and you get a yellow precipitate. So this idea of butter, eggs, and sugar. Butter, eggs, and sugar. So let's just go through that again so we are 100% sure of these ions here. And all of these ions and all of the tests. So in order for testing for gases, so we want to be able to test for chlorine gas, for oxygen, hydrogen, and carbon dioxide. So they're the ones we'll be testing for. And then what I want to do is I'm going to put the test and then the result. So for chlorine, we use the test of the damp litmus paper, and it should be bleached white. For oxygen, we use the splint in the test tube of oxygen, and it should relight if it exists. Then we have hydrogen. So for hydrogen, we will have the squeaky pop test. So you have a dimly lit splint right near the hydrogen test tube, and it should make a noise. And then you have carbon dioxide, which is the lime water test. It should turn cloudy. Now they're the gases tests. Now we also need to know the anions tests. So which anions do we have to remember? We have to remember carbonate, sulfates, carbonate, sulfates, and halides. So carbonates are tested with dilute acids. And what happens here is the carbonate reacts with the dilute acid and produces carbon dioxide, which turns lime water cloudy. We have sulfates. Now the test for sulfate ions is reacting with hydrochloric acid. So instead of dilute acid, sulfate's hydrochloric acid. And then what happens is you also add barium chloride. And then what happens is the barium chloride will form a white precipitate. And finally, for halides, you react it with nitrates, nitrogen, nitric acid, sorry, because these are all to do with acids. So we have nitric acid, as well as some silver nitrate, and they should then form the various colours that I'm going to look at here. So if we have silver and chlorine, if we have silver and bromine, if we have silver and iodine. So if we have silver chlorine, we get silver chloride, which will be a yellow, a white colour, silver chloride. If we react silver and bromine, we get silver bromide, which is a cream colour. And then if we get silver and iodine, we get silver iodide, which is a yellow colour. We've been working for 2 hours, and so far we've covered 2 biology topics, and we're on our second chemistry topic, so we're doing quite well. Okay, so that's gases and that's anions revised. Now let's look at the tests for cations. Okay. So compounds of some metals burn with a characteristic colour, so they have a distinctive colour flame. So we have lithium, sodium, potassium, calcium, and copper. So lithium, sodium, potassium, calcium, copper, and they all burn with their distinctive flames. So lithium has red, sodium has an orangey yellow, Potassium has the lilac flame. That's not quite lilac, is it? That's more of a lilac. Yeah. Potassium has lilac. Calcium has this orange, red, dark and dull flame like this. And then copper has this green flame, which is really pretty. So these are the flames that we have. So lithium is red, sodium is orange, potassium is a lilac flame, then calcium is a burnt orange flame, and then copper is a green flame. So all you have to do is you have to get a platinum wire loop, dip it in hydrochloric acid. So you get your wire loop, dip in hydrochloric acid, and then hold it in a blue flame on the Bunsen burner. And you're then going to record the colour of the flame. Now you can use these colours to detect and identify different ions. However, it only works for samples that contain a single metal ion, because if it contains a mixture, then the flame colours can be masked and hidden. Now, some metals we can use by looking at how it forms coloured precipitates with sodium hydroxide. So many metal hydroxides are insoluble, therefore they can't dissolve water and precipitate out of solution when formed. So some of these hydroxides have a characteristic colour. So metal hydroxides, a lot of them are insoluble, which means that if we react them within a solution, and because of many of them have a characteristic colour, we should be able to identify which metal hydroxide it is. So let's look at some examples. So we had calcium, and calcium is an insoluble metal hydroxide, and therefore forms the precipitate of the white colour when it reacts with anything. The ionic equation for the precipitate formula would look like this. So calcium plus the OH creates calcium hydroxide. So our aim in this is to change the metals to metal hydroxides by reacting them with oxygen and hydrogen, and therefore you will get its colours. So your calcium turns white, copper turns blue, despite its green flame, iron 2 plus and iron 3 plus. So iron 2 plus turns green, and iron 3 plus turns brown. Aluminium, white is initially white, but then redissolves in excess to form a colourless. So then in excess it's colourless. And then we have magnesium. Magnesium has a 2 plus, and then again is white. So we have copper, which turns blue, iron 2 plus, which turns green, iron 3 plus, which turns brown, aluminium 3 plus, which turns white and then colourless, and then magnesium, which again turns white. Now let's look at flame emissions spectroscopy. Every metal ion gives a characteristic line spectrum. Okay, every single metal ion. So during flame emission spectroscopy, a sample is placed in a flame. Their electrons become excited. When the electrons drop back down to their original energy levels, they then transfer their energy as light. So what happens is, when we have ions that are placed within a flame, these ions then get excited. with all of the high energy. Then when the ions drop back levels, what happens is they transfer this energy as light. Now the light then passes through a spectroscope, which can detect different wavelengths of light to produce a line spectrum. Now the combinations of wavelengths emitted by an ion depends on its charge and its electron arrangement. Now, since no two ions have the same charge and the same electron arrangement, different ions then emit different wavelengths of light. So each ion produces a different pattern of wavelengths and has a different line spectrum. The intensity of the spectrum indicates the concentration of that ion in solution. This means that the line spectrums can be used to identify ions. This is a relatively basic notion. We then have to quickly revise evolution of the atmosphere and chemistry of the atmosphere. I think the major thing, it's literally 3 pages, so it's not too much to revise. And the major thing that we need to understand about chemistry of the atmosphere is what the atmosphere was like before and why it was like this. So we have 3 phases: phase 1, phase 2, and phase 3. We're in phase 3 right now, but we need to know about phase 1 and 2. So phase 1 is volcanoes giving out gases. Volcanoes gave out gases. The surface was covered in volcanoes the first billion years of Earth's history, and they erupted and released lots of gases. The early atmosphere was probably mostly carbon dioxide. So mostly carbon dioxide and also virtually no oxygen. This is quite like the atmospheres of Mars or Venus. Now, volcanic activity also released nitrogen. Now, this nitrogen built up in the atmosphere, as well as water vapor, and small amounts of methane and ammonia. So volcanoes, through their eruption, they mainly release carbon dioxide and nitrogen. There will be no oxygen, but there's also tiny signs of water vapor, methane, and ammonia. Phase 2 are the oceans, algae, and green plants. So, as just mentioned, the oceans, algae, and green plants began to then absorb this carbon dioxide. So where the water vapor in the atmosphere condensed, it formed oceans. And lots of carbon dioxide was removed from the early atmosphere as it had dissolved in the oceans. This dissolved carbon dioxide went through a series of reactions to form carbonate precipitates that formed sediments. So the oceans were formed by lots of the water vapor from the volcanoes and where it had settled. And also in oceans, we have lots of carbon dioxide and carbon precipitates because they went through a series of reactions such as sedimentation. The green plants and algae evolved and absorbed some of the carbon dioxide so that they can carry out photosynthesis. And then what happens is some of the carbon these organisms took in from the atmosphere became locked up in rocks and fossil fuels. And phase 3 are green plants and algae and how they produced oxygen. As well as absorbing carbon dioxide, they release oxygen. Now, algae evolved first around 2.7 billion years ago, and then over the next billion years or so, green plants evolved. Now, as oxygen levels built up in the atmosphere, the more complex life could then evolve. So around 200 million years ago, the atmosphere reached a composition similar to what it is today. So 200 million years ago, which is roughly 80% nitrogen, 20% oxygen, and small amounts of other gases. Okay, so that is chemistry of the atmosphere revised. Just take a 1 or 2 minute break away from the screen because it's now been just over 2 hours. For the final topic that we're going to be revising for this last 12 minutes, we'll be going over using resources, and I'm just going to voice this topic so you can hear it and reflect back on it and just focus on this rather than having to look at the screen, because I know it's a bit strenuous to constantly look at the screen. So let's begin now. Using resources chemistry revision. Ceramics. Ceramics are non-metal solids with very high melting points. They aren't made from carbon-based compounds. Some ceramics can be made from clay. Clay is a soft material When it is dug up out of the ground, it can be molded into different shapes, and when fired at high temperatures, it hardens to form a clay ceramic. Another example of a ceramic is glass. Glass and can be brittle when thin. Most glass is soda lime glass, which is made by heating a mixture of limestone, sand, and sodium carbonate until it melts. When the mixture cools, it comes out as glass. Borosilicate glass has a higher melting point than soda lime glass. It's made in the same way as sodium lime glass, using a mixture of sand and boron trioxide. Composites are made of one material embedded into another. Fibers or fragments of a material, known as the reinforcement, surrounded by a acting. Properties of a composite depend on the properties of the material it is made from. For example, let's look at fiberglass. Fiberglass contains fibers of glass embedded in a matrix made of polymer. It has a low density, but it's very strong. It's used for things like skis, boats, and surfboards. Carbon fiber also has a polymer matrix. The reinforcement is either made from long chains of carbon atoms bonded together, carbon fibers, or from carbon nanotubes. These composites are very strong and light, so are used in aerospace and sports car manufacturing. Concrete is made from aggregate, which is a mixture of sand and gravel embedded in cement. It's very strong, which makes it ideal for use as a building material. Wood is also a natural composite of cellulose fibers held together by an organic polymer matrix. Now, polymers can have very different properties. Two important things that can influence the properties of a polymer is one, how it's made, and two, what it's made from. For example, the properties of polyethylene depend on the catalyst that was used and the reaction conditions, meaning the temperature and the pressure that it was created with. Low-density polyethylene is made from ethylene at a moderate temperature under a high pressure and with a catalyst. It's flexible and is used for bags and bottles. High-density polyethylene is made from ethylene but at a lower temperature and pressure with a different catalyst. It is more rigid and is used for water tanks and drain pipes. The monomers that a polymer is made from determines the bonds that form between the polymer chains. These weak bonds between the different molecule chains determine the properties. So we have two different types of polymers: thermosetting polymers and thermoforming polymers. Now thermosoftening polymers, and thermosetting. So thermosoftening polymers contain individual polymer chains entwined together with weak forces between the chains. You can think of them as spaghetti, very weakly. You can easily melt these plastics and then remold them, therefore they're recyclable. However, thermosetting polymers contain monomers that form crosslinks between the polymer chains. These then hold the polymers together in a solid structure and prevent it from melting under heat. Therefore, they will just char and burn when heated instead of melting. Now, it's important to note how different materials are suited to different jobs. What materials are used for depends on their properties, and you need to be able to interpret information about the properties of materials and decide how suitable they would be for different purposes. Ceramics include glass and clay ceramics, such as porcelain and bricks. They're used for insulators as heat because they're quite brittle, but they aren't very flexible. Polymers are used as insulators for heat and electricity. They can be flexible and they are easily molded, especially thermosoftening polymers, and they have many applications, including clothing and insulators. The properties of composites depend upon the matrix binder and the reinforcement used to make them, so they have many different uses. Metals are malleable, good conductors of heat and electricity, and are ductile. They are shiny and stiff, and they have many uses, including in electrical wires, car bodywork, and cutlery. Now, pure metals don't always have the properties needed. Therefore, we make alloys by adding another element to the metal. This disrupts the structure of the metal that was previously organized and regular, and therefore distorts it, making alloys harder than pure metals. The alloys of iron are called steels and are often used instead of pure iron. So we have low carbon steel, which is roughly 0.3% carbon. It's easily shaped and it's used for car bodies. High carbon steel, which is roughly 3% carbon, and is very, very strong, but it's more brittle. And this can be hardened. And stainless steel, which is got 11.5% chromium, is corrosion resistant and it's used for cutlery. Now, many other alloys are used in everyday life. So bronze is made from copper and tin. Bronze is actually harder than copper and is used to make medals and decorative things. Brass is made from copper and zinc. Brass is more malleable than bronze and is used in situations where lower friction is required, such as water taps and instruments. Now, gold alloys are used to make jewelry. Because pure gold is very soft, metals such as zinc, copper, and silver are used to harden it. Pure gold is described as 24 carat, so 18 carats means that 18 out of 24 parts of the alloy are gold. And aluminium alloys are used to make aircraft, because aluminium is one of the most commonly occurring elements on the planet, and it has a low density, which is an important property in aircraft. But pure aluminium is too soft for making aeroplanes, so therefore it's alloyed with small amounts of metals to make it a bit stronger. Now, iron and steel corrode much more than aluminium. So corrosion is just when metals react with substances in their environment and are gradually destroyed. Now, iron corrodes easily. In other words, iron rusts. The word rust is only used for corrosion of iron and no other. Now, in order to rust iron, it needs to be in contact with both oxygen and water, because the chemical equation for rust for iron is iron plus oxygen plus water creates hydrated iron oxide. Now, corrosion only happens on the surface of a material when it's exposed to the air. However, rust is often crumbly and flakes off, leaving more iron to rust again. This means that eventually all the iron will be object to corrosion. Now, aluminium also corrodes. However, aluminium isn't completely destroyed because aluminium oxide forms a thin layer around it, which actually protects it and is also invisible from the human eye. Now, both air and water are needed for iron to rust, and we can do chemical practicals to support this. There are two main ways to prevent rusting. Firstly, you can coat the iron to create a barrier. So, painting or coating with a plastic. Electroplating, which is using electrolysis to reduce a metal ion onto an iron electrode, so it can be used to coat the iron with a layer of metal that won't corrode. And oiling and greasing. This is used when moving parts are involved, like bike chains. Another method is a sacrificial method, placing a more reactive metal, such as zinc with iron, so that the water and oxygen will actually react with the zinc, instead protecting the iron. Now, an object can also be galvanized by spraying a coating of zinc onto it. The zinc is firstly protective, but if scratched, the zinc site around the scratch works as a sacrificial method. So this is a combination of both. Now, we have different resources. Natural resources come from the earth, sea, and air. Natural resources form without human input. Things like rubber coming from the sap of a tree. But polymers are man-made. Now, renewable resources. So, extracting finite resources has their many risks, including even the risks in jobs. Reusing and recycling is really important for sustainability. Copper is a finite resource. One way to improve copper's sustainability is by extracting it from a low-grade ore, which means ores without much copper in it. Scientists are looking into ways of doing this. So there's two ways: bioleaching and phytomining. Bioleaching is when bacteria is used to convert copper compounds in the ore into soluble copper compounds, separating out copper from the ore in the process. So bioleaching is when bacteria converts copper in the ore into soluble copper compounds. Now the leachate, which is the solution produced in the process, contains copper ions which can be extracted by electrolysis or displacement. Phytomining. Now phytomining involves growing plants in the soil that contain copper. The plants can't use or get rid of this copper, so it just gradually builds up in the leaves, and then this can be harvested, dried and burned in the furnace. Now the ash contains soluble copper, which copper can be extracted by electrolysis. Experiments to these new methods have a smaller impact. Now mining and extracting metals take in a lot of energy, most of which comes from burning fossil fuels, whereas recycling metals often uses much less energy than is needed, and then to extract a new metal and conserves the finite resource of each metal and cuts down the amount of waste getting sent to landfill. Metals are usually recycled by melting them and then casting them into the shape of the new product. Depending on what the metal will be used for after recycling, the amount of separation for the recyclable materials can change. Glass recycling can help sustainability by reducing the amount of energy to make new glass products, and also the amount of waste when glass is thrown away. Now we're going to look at life cycle assessments. So life cycle assessments, potable water, treatment, Haber process, and PK fertilizers. Life cycle assessment. This looks at the total environmental cost. It looks at every stage of a product's life to assess the impact it would have on the environment. One, it looks at getting the raw materials because it can damage the local environment and it needs to be processed. Two is manufacturing and packaging. Manufacturing products and the packaging uses a lot of energy resources, causes a lot of pollution, and you also need to think about any waste products and how to dispose of them. Three, using the product. The use of a product can damage the environment. For example, burning fossil fuels releases greenhouse gases and other harmful substances. How long? A product is often useful, and how many uses is also important. And finally, product disposal. Is it disposed onto landfill sites, and does it pollute land or water? Energy is used to transport it to landfill. Are the products incinerated using fire energy? You can then compare the life cycle assessments. However, there are problems with life cycle assessments. For example, the use of energy, some natural resources, and the amount of certain types of waste produced by a product over its lifetime can easily be quantified, but some things are harder, such as the effect of pollutants giving a numerical value. So producing a life cycle assessment is not an objective method, as it takes into account the values of the person carrying out the experiment. So therefore it can be biased. Also, selective life cycle assessments, which only show some of the impacts of a product. This can also be biased. Now, potable water is water that has been treated and is naturally safe for humans to drink and is essential for life. Chemists wouldn't call it pure though, because pure water only contains H2O molecules. The important thing is that the levels of dissolved salts aren't too high, that it has pH between 6.5 and 8.5, and finally that there aren't any bacteria or microbes. Now, the way that potable water is produced depends on where you are. So rainwater is a type of fresh water. Fresh water is water that doesn't have much dissolved in it. So when it rains, water can either be collected as surface water in lakes, rivers and reservoirs, or as groundwater in rocks called aquifers that trap water underground. In the UK, the sources of fresh water usually depend on the location. Surface water tends to dry up first, so in warm areas such as the southeast, most of the domestic water actually comes from groundwater in northern areas. though it only has low levels of dissolved substances, water from fresh water sources still need to be treated. So firstly, it goes through filtration through a wire mesh that screens out large twigs and gravel, and then we have a gravel and sand bed. This filters out solid bits. And finally, sterilization to kill any harmful bacteria or microbes, which is usually done by bubbling chlorine gas through it or using ozone or ultraviolet light. In some very dry countries, for example Kuwait, there's not enough surface or groundwater, so seawater must instead be desalinated. Distillation can be used to desalinate seawater. So first you have to test the pH. Here's how you can test and distill water. First test the pH using a pH meter, then test the water for the presence of sodium chloride, which is the main salt in seawater. Then test for sodium ions by using a flame test, and it should be yellow. Then test for chloride ions by adding dilute nitric acid and then silver nitrate solution, and it should turn white. Then to distill the water, pour the salty water into a distillation apparatus and heat the flask below. The water will boil and form steam, leaving any dissolved salts in the flask. The steam will then condense back to liquid water in the condenser and be collected as it runs out. Then you want to retest the distilled water for the presence of sodium chloride and check that it has been removed. Seawater can also be treated by processes using membranes, such as reverse osmosis, where the salty water is passed through a membrane that only allows water molecules to pass through, and ions and larger molecules are trapped and separated from the water. However, both distillation and reverse osmosis need loads of energy, so they're really expensive and not really practical. Now we're going to look at wastewater treatment. Now, wastewater comes from loads of different sources. We use the water for lots of things at home, like having a bath, going to the toilet, or doing the washing up. When you flush this water down the drain, it goes to the sewers and towards sewage treatment plants. Agricultural systems produce a lot of wastewater as well, such as nutrient runoff from fields and slurry from animal farms. Sewage from domestic or agricultural sources then has to be treated to remove any organic matter and harmful microbes before it can be put back into freshwater surfaces like rivers or lakes. Otherwise, it would make them very polluted. Industrial processes, such as the Haber process, also produces a lot of wastewater that has to be collected and treated. As well as organic matter, industrial waste can also contain harmful chemicals, so it has to undergo additional stages of treatment. So, before being treated, the sewage is screened. This involves removing bits of bags, grit, etc. Then it's allowed to stand in a settlement tank, undergoing sedimentation. So the heavier particles sink to the bottom and form a sludge, which is produced, while the lighter, effluent particles float on the top. The effluent particles in the sediment tank are removed and treated by biological aerobic respiration, where air is pumped through the water to encourage aerobic bacteria to break down the organic matter, and then the sludge from the bottom of the sediment is removed and transferred into large tanks where it's broken down by a process called anaerobic digestion. Now, anaerobic digestion breaks down the organic matter in the sludge, releasing methane gas in the process. Now, the methane gas can be used as an energy source, and the remaining digested waste can be used as fertilizer. For waste containing toxic substances, additional stages of treatment may involve adding chemicals, UV radiation, or an additional membrane. Sewage treatment requires more processing than treating fresh water, but uses less energy than desalination or salt water, so it could be an alternative in areas where there's not much fresh water, like Singapore. And finally, we're going to look at the Haber process and NPK fertilizers. So the Haber process is used to make ammonia from hydrogen and nitrogen using the following reaction. Nitrogen plus hydrogen creates ammonia and heat. This reaction is well suited for an industrial scale, as the reactants nitrogen and hydrogen are not difficult or expensive. Now, the nitrogen is easily obtained from the air, being 78% nitrogen. The hydrogen comes from reacting methane with steam to create hydrogen and carbon dioxide. Now, the reactant gases are then passed over an iron catalyst. A high temperature of 450 degrees and a high pressure of 200 atmospheres is used. Now, because the reaction is reversible, it occurs in both directions. Some of the ammonia produced converts back into hydrogen and nitrogen in dynamic equilibrium. Now, the ammonia is formed as a gas, but it then cools in a condenser and liquefies and can be removed. The unused hydrogen and nitrogen are then recycled, so nothing is wasted. The ammonia produced can then be used to make ammonia nitrate, a very nitrate-rich fertilizer. Now, as we said, the reaction is reversible, so there's a compromise to be made. So, the position of equilibrium for a reversible reaction, there's a trade-off between increasing the rate and maximizing the yield. For the forward reaction in the Haber process is exothermic. This means that increasing the temperature will move the equilibrium the wrong way. Therefore, the yield would be greater at lower temperatures. The trouble is, lower temperatures means a slower rate of reaction, and the 450 degrees Celsius is therefore a compromise between maximizing the yield and maximizing the speed of the reaction. Higher pressure moves the position of equilibrium towards a product, since there are 4 moles on the left-hand side for every 2 moles on the right. Therefore, increasing the pressure increases the yield, which also increases the rate of the reaction. So pressure is therefore set as high as possible, which is 200 atmospheres. And finally, the iron catalyst also makes the reaction go faster without being used up. And finally, NPK fertilizers. Farmers used to use manure to fertilize fields. Formulated fertilizers are much better, however, because they're more widely available, they're also easier to use, and they don't smell because they'd have just enough of each nutrient, so more crops can be grown. Now, the 3 main essential elements in fertilizers are nitrogen, phosphorus, and potassium, which is where we get the NPK. Nitrogen, phosphorus of these elements, their growth and their life processes are affected. So these elements may be missing from the soil if they've been used up by a previous crop. Fertilizers then replace these missing elements to provide more of them. For example, fertilizers add more nitrogen for plants to produce proteins. So NPK fertilizers are formulations containing salts of nitrogen, phosphorus, and potassium in the right percentages of the elements needed. Now, ammonia can be reacted with oxygen and water in a series of reactions to make nitric acid. You can also react ammonia with acids, including nitric acid, to get ammonium salts. Ammonia and nitric acid react together to produce ammonium nitrate, which is especially good compound to use for fertilizer because it has nitrogen from 2 sources. So ammonia plus nitric acid creates ammonium nitrate. This reaction is carried out in different ways. You may be asked to compare the differences between the different metals. So in the industry, the reaction with ammonia plus nitric acid makes ammonium nitrate is carried out in giant vats at high concentrations in a very exothermic reaction. The heat released is used to evaporate water. In the lab, the reaction is carried out using a smaller scale of titration and crystallization. The reactions are at a much lower concentration than industry, so less heat is produced, and it's safer for a person to carry out. After the titration, the mixture then needs to be crystallized to give pure ammonium nitrate crystals. Now we've got the ammonium nitrate, but what about the phosphate and potassium? These are then sourced from mixed compounds. They are mined and used as a source of potassium. Phosphate rock is also mined. However, because the phosphate salts in the rock are insoluble, plants can't use different acids produces soluble phosphates. So reactions with nitric acid produces phosphoric acid. Reactions with sulfuric acid produces calcium phosphates. And reactions with phosphoric acid only produces calcium phosphate, and there will be triple superphosphate. And that's it for GCSE Chemistry, Topic 10. So now we are going to move on to physics. Hello everyone! So, welcome back. It's a final push till we get through physics. We're just going to go over waves and then magnetism and electromagnetism. I have quite a fun way of teaching you that. And then we're going to go over space. So, only the fun topics left, I guess. So, let's move on to physics. Sorry for this. Okay, let me share the screen now. So, yes, as I said, first we're going to be going over waves. So there are two different types of waves that we see. There's transverse and then there's longitudinal. So let's describe themselves. So transverse waves and longitudinal. So transverse waves are when the oscillations are perpendicular to the direction of the energy transfer. So let's just say oscillations perpendicular to the direction of the energy transfer. Longitudinal therefore means the oscillations are parallel to the direction of the energy transfer. So let's look at what this would mean then. Transverse, so this is the direction of the energy transfer, so the oscillations are therefore moving perpendicular to the direction of the energy transfer, whereas longitudinal is moving parallel to the direction of the energy transfer. So transverse waves are things such as kinetic energy or light waves. And longitudinal is something like sound energy. Now, all longitudinal waves require a medium. So sound requires a medium to travel in. Sound does not travel in space because there are no particles, and this is because longitudinal requires the vibrational particles to transfer energy. However, not all transverse waves require a medium. For example, light. Light waves do not require a medium, and we know this because that is exactly how the sun transfers its light to Earth, because we know that this space in between is a vacuum. However, the light still transfers its energy. Now we need to know properties of waves. So when we're looking at a wave, we have different properties. So this is called the peak. This is called the trough. The amplitude is the displacement, the maximum displacement from the undisturbed position. So amplitude would look like either this or this length. That's amplitude. Now we have values such as frequency and wavelength. So wavelength is the length of a wave from one point, so let's say from this peak here to the next peak. So that's a wavelength. And it's represented using this interesting symbol that looks like that. And then we have the frequency. So the frequency is the number of waves that pass a point per second. So let's say the number of waves that pass this point per second. And one wave, as we said, is viewed by this metric here of one wavelength. And the period is the time it takes for one wave to pass. And therefore, we get the equation period is equal to 1 over frequency, which you'll be given in your exam. Now, when electromagnetic waves are outputted, changes take place in the atoms. So what happens is we fire electromagnetic something. What we're doing is there's changes that take place in the nuclei of the atoms, which we looked at in the radiation topic. So what happens is the electrons can move from a lower energy level to an outer energy level, it then releases all of its radiation and then returns back. And it does this to try and stabilize. This is similar to radioactive decay. Now, gamma rays can be emitted from the nucleus, which is represented by the symbol Y. That's gamma rays. So when electromagnetic waves are absorbed, they can also cause changes to atoms, and they can be absorbed by a large range of radio waves. So hazards of electromagnetic waves are things like how ultraviolet, so UV, X-rays, and gamma rays all have the potential of being hazardous to the body. And the dose of the radiation used is measured in something called sieverts. Now, radio waves can be produced when electrons oscillate in electric circuits. So, radio waves are produced in electric circuits because in circuits we see that similarly the electrons oscillate. Now, they can also be absorbed, which causes electrons to create alternating current. So when electrons oscillate, it then creates the alternating current frequency. Now, it's important to know this wave equation when going back to the properties of a wave. So the wave equation is the wave speed is equal to the frequency multiplied by the wavelength. Wave speed is frequency times wavelength. Wave speed is the speed at which the wave moves through a medium. So there's actually 2 required practicals in this topic that are quite highly important that you'll need to know. So the first required practical is the ripple tank one. So the ripple tank is when there's a ripple tank used to observe the features of waves. So in a ripple tank, the ripple tank is used to observe the features of the waves. And what happens is there's a vibrating bar which is connected to a power pack. Which causes waves to happen, and then you can shine a light which can allow you to get a drawing of it and then measure certain properties of the waves. Now, this is waves in a liquid, but there's another required practical regarding waves in a solid. So when we're looking at waves in a solid, you start with string, which is attached to a vibration generator, and this then vibrates. And at the other end is a hanging mass. What happens is a vibration generator is then attached to a signal generator, which changes the frequency. When you power it on, the string vibrates. Now, the standing wave due to an effect is called resonance. So this will be referred to as the resonance. The standing wave, although mostly it will be really difficult to see, it's constantly moving like that. You can measure things like wave speed by looking at the wavelength using a ruler and the frequency and calculating wave speed. So here we're calculating wave speed. In the other instance, we were calculating and being able to draw a graph and profile of the wave. Now we're going to look at reflection and refraction of waves. So, reflection. Reflections of waves happen when waves hit. So when a wave hits a boundary, a couple of things can happen. What happens? Well, let's see. The first thing that can happen is it can just transmit straight through. Second thing is it can be absorbed by the medium. It could be reflected, and it can be refracted. Okay, so normally if it's a mirror is shown like this. So if it was a mirror, it would just reflect. Now if it was an uneven surface, then it will be shatter and produce a shattered image, which is called refraction. So sometimes you may be asked in an exam, so you have the mirror here, and let's say you have an object. And you want to see where the object will reflect to. Then in order to do that, normal in the middle, and then draw a line from all those points like that. These are the lines. And then we know that's going to produce a reflected image like this. And therefore, you'll get the image, the reflected image. Now we also have refractions of waves. So waves can change direction when they hit a boundary as well. So refraction, this is more diffuse reflection and specular diffuse reflection. But we can also note how refraction can happen in another way too. So refraction can happen in terms of when we're looking at the reason why refraction happens and bends towards the normal is because of the fact that when it's entering from a gas to a solid, it slows down because it takes longer to transmit, and therefore because it slows down, it winds towards the normal. But then when it leaves, it goes back against normal because it's much faster. And that's why in the water, sometimes when you put your hand in, it looks like your hand is over there. And you're confused, but the reason why is because the image takes longer to appear in the liquid than as it does in the gas of air. And that's why it appears that way. There's then another required practical about reflection and refraction, which is all about this glass block here. Okay. Now we're looking at ultrasound and seismic waves. So ultrasound is a type of electromagnetic wave, and it's actually a type of sound wave, believe it or not. The frequency is much higher than human hearing, so we are unable to hear it, but some animals are, like dogs. So it's higher than the upper limit of hearing. Now, ultrasound partially reflects at the back. Ultrasound is not only a sound wave, but it also is a wave that reflects at the boundary between two different mediums. Now, that means that it can then be used to calculate things like distances and produce internal images. And the good thing is that there's no mutation risks and that there's no causes of cancer, therefore. So this is quite a safe route. Now, determining the distance using ultrasound, we can use the equation distance is equal to speed multiplied by time. Now we've looked at ultrasound, it's also important to look at seismic waves. Seismic waves are these massive movements that occur due to tectonic plate movements. So they carry, what happened, their whole purpose is carrying energy and dispersing energy away from the earthquake. And what happens is as they pass through the layers of the earth, and they can be detected by seismometers. Which we found out the structure of the earth. So the seismometers look at the earth, and we know that the structure, and we've come up with this structure here of how we have the inner core, the outer core, the mantle, and the crust. We've come up with this based on the seismic waves. So what we have is we have P waves, and we have S waves. So P waves, and the patterns of these waves depict the interior structure of the Earth. So P waves are longitudinal. And remember, we looked at what longitudinal are. So longitudinal are the ones which are parallel to the direction of energy's transfer. So they're like sound, like that. So the P waves, we looked at the P waves look like this. So the P waves tend to ignore the core. And then in this area, I'll use a brighter pink to show that this is what we would call a P wave shadow zone. Now, P waves being longitudinal and going through these parts tells us that because P waves need a medium in order to transfer, it tells us about the fact that some areas are denser than others. So the P wave shadow zone told us that these must be liquid because sound is hard to transfer during liquid. So these areas must be liquid. And then we looked at the S waves. So the S waves, let's present the S waves with longitudinal. They are transverse. And the S waves went here and here, but were not detected anywhere else. So there's this massive S wave shadow zone. And this S wave shadow zone suggests that it can't pass through liquid. So this type of transverse wave can't pass through liquid. So both of these waves showed us that this side is liquid. Okay, we're now going to go over more about electromagnetic waves, and then we'll talk about light and ray diagrams. So let's look at electromagnetic waves. So light is an example of an electromagnetic wave because it's transverse, so it transfers its energy from a source to the absorber. So electromagnetic waves are waves that transfer energy from a source to an absorber. For example, we looked at light and how light transfers energy from a source to the absorber. Now, if we pass white light through a prism, it splits. Into a spectrum. Now, each color has a different wavelength, and then etc. So we've got red, orange, yellow, green, blue, indigo, and violet. So in terms of what each one has, red has a lower frequency, but has a longer wavelength. So we'll look like this. Whereas violet, so right at the end, so violet has the highest frequency and the highest wavelength. Oh, the highest frequency and the shortest wavelength. So it'll just be like this. And that's how it moves. So red is nice and relaxed, and violet is like uptight. Now we can use this same idea with radiation. So we've got radio waves, microwaves, infrared waves, visible light, ultraviolet, X-ray, and gamma. So they're the waves. So we have a low frequency, a large wavelength, and then the frequency starts to increase. You go like that. So as you go this way, the frequency increases, but the wavelength decreases. So the electromagnetic spectrum is just a continuous spectrum, and it means that the cutoff point between one wave and another wave is not always clear. Now, a way to remember these letters are, you can say raw meat is very unsanitary except giraffe, which might help you remember it. So we've got radio, microwaves, infrared, visible light, ultraviolet light, we'll say except giraffe, X-ray, and gamma ray. So all electromagnetic waves travel at the same speed in a vacuum. We looked at this in another concept in physics. So all electromagnetic waves travel at the same speed. Just how, if you look at in a vacuum, all things would fall down at the same speed as well. So if you had a ton of bricks, if you had a massive ton of stuff, and if you had a feather, in a vacuum, for example, on the moon, this would fall down at the same exact rate, at the same exact speed as that. And that's why in a vacuum, even electromagnetic radiation all travels at the same speed. We then have a required practical, which is about infrared, and we're trying to see how much infrared is emitted or absorbed by different surfaces using Leslie's cube. Now, Leslie's cube has different surfaces on different materials on each surface. and each surface will therefore, you can then point an infrared detector at it. So imagine pointing a laser at it. You have to keep the same distance, so same distance, keep that an independent variable. And what you're going to do is you're going to, on the other side, you're going to have Vaseline and a drawing pin. You're going to see how long it takes for that Vaseline to fall down. And finally, on electromagnetic waves, what are some of the uses of electromagnetic waves? Well, radio waves are used in TV signals because it can travel a long distance, as well as microwaves. And then microwaves are used in microwave ovens, as well as again TV signals because it can travel long distances. Infrared is used in electrical heaters and cookers. We have visible light. Now, visible light is used in communications of fiber optics. In which thin strands of glass are used. And then ultraviolet light is used for like sun tanning, energy efficient light bulbs, and then X-rays and gamma rays are used for medical imaging. X-rays are used for bones, and gamma rays are used for like cancers. And they're both very penetrative. And finally, just doing some revision on glass. On glass, I mean light. So we have different types of lenses: convex lens and a concave lens, because it caves in. So concave and convex lenses. Now convex lenses, that's good. Convex lenses, all concave lenses all diverge into different directions. Now, convex lenses are represented by this, and convex lenses are represented by this. So when we have from one focusing point and two focusing points, we have different images. So for a convex lens, if the object was two focusing points away, then what you would do is you draw a line from the top of the focusing point, top of the object, through the center, and then you draw a line from the top of the object through the axis, and then so let's do this again. Let's do it here. So we have, okay. So if it was two focusing lenses away the object, what you do is draw the first straight through the center, or around the center, and then where, actually that doesn't make sense, does it? And then we've got the object. So you draw a line from top. Oh, so that's how you do it. So first you draw a line from the top of the object, and then you draw a line from here, and then wherever those two meet is where the image is. So in this case, for a convex lens, the image is smaller. It's formed upside down, so it's inverted. So smaller is what we call diminished. So the image is diminished, it's inverted, and it's a real image. The reason why it's a real image is because the rays actually meet at a point. Now, the closer the object is, then the less focusing there is. So imagine, let's say the object was closer, 1 focus away. Then again, you draw your line from here, and you've got a line from there, and you've got a line from there. Again, their points meet. So here, the image is larger than the object because the image is larger than the object. So if it's more than 2 focusing lenses away, then it's smaller. If it's is 1 focusing lens, then the image is larger, but it's still inverted and it's still a real image because it reaches a point. Now, concave lenses are different. So concave lenses, say if you had a point here, and you've got 1 focusing lens, 2 focusing lenses. So what you would do, say the object is here, then it's going to be like that, and it's diminished and the right way up. And imagine that's there, and then you're going straight down like that as well. And you see that it does not meet. So when it is between 1 focusing lens and 2 focusing lenses, it's not a real image, it's magnified, and it's the same way up as the object. So the not real image is called virtual. Black body radiation is when matte black surfaces absorb everything. Visible light. So we have specular, like so. That's nice and specular. And diffuse is scattered because of the rough surface, which scatters light rays. Okay, and finally, second to finally, we're looking at electromagnetism now. Now, I have a good way of revising electromagnetism here. So let me just get rid of this. Okay. Electromagnetism. So this is how I revised it, which I found good. So firstly, whenever there is a current carrying wire, which in this case we're going to be represented by brown, because brown represents live wire. So whenever there's a current carrying wire, then it generates a magnetic field. Now, due to the right-hand Fleming rule, which you have your thumb, and then you have your hand, and then you have your fingers, which are going this way. So this then suggests your thumb will be the direction of the wire, current carrying wire. And then your fingers will be the direction of the magnetic. We see that the magnetic field is going this way. And a current carrying wire therefore becomes an electromagnet. Now, we have the idea of what is a solenoid. So here we also have something called a coil. Now a coil, let's introduce coil as coil sing. That's a coil, okay? Now this is coil sing. Imagine as a person. And this coil generates a magnetic field. Of the magnetic field. Just note that the magnetic field within the coil is uniform. But outside of the coil is just spaced out and at different points. But within it, it's this, the strength and the direction is the same at every point. So we've introduced here to a solenoid then, because a solenoid is when a current carrying wire is made into a coil. So you've got a solenoid here, which is a current carrying coil of wire. Right? Which also generates a magnetic field. Now, coil sing then wants to get married to iron core, which is represented by blue. So here we have iron core, right? And they want to get married, and what happens is once they get married, they then form an electromagnet. He protects her on the outside. So we have the solenoid here, which is a current carrying coil of wire around the iron core. So the iron core then becomes induced. She becomes induced, i.e. you could imagine her as like pregnant because she's just having a kid. So she becomes induced after they get married. And this then produces an even stronger magnetic field. And that's how electromagnets work. Now, electromagnets can be really, really useful. And the reason why I say that is if you look at the diagram that I'm going to draw now. So, say we have the iron core, and then we have the coil sing. And then this is then connected to a power source, and is then connected to a switch. Now imagine this is the iron contact, okay? This is the husband. The husband's on the left side. So this is actually just a coil of wire. So the husband's on the left side and the wife is on the right side. The coil and the iron contact. And then the iron contact then is connected to a striker that will strike the bell. So when the switch is then turned on, we then see current flow it then induces the coils. The coil then becomes a current carrying wire and then gets a magnetic field. But then the current also goes towards this, and then this magnetic field then induces the ion contact. So the ion contact is then it wants to go towards the spring, so it moves towards it and then strikes the bell. And then as this strikes the bell, it then breaks the switch and it just removes back to where it was. And that's all you need to know about electromagnets and how they can be useful. Now, we're going to look at the motor effect. So the motor effect looks like this. So what happens is when at right angles, the motor effect is basically when two magnetic fields come into contact, and it's just how they act with each other. So not only do we have the electromagnet, which is, again, we have the current carrying coil of wire that's produced this way. So we have the current carrying coil of wire, and then we have the two magnets. So we have the north pole, and then we have the south pole. So here, this brown wire is current carrying, so therefore it's induced a magnetic field, and because it's going in this direction, the magnetic field goes in this direction. So we then have this diagram here. But then there's also a magnetic field between the north to south. So let me portray this in a darker green, going from this way to this way. Now that means that the magnetic fields are now interacting. And what happens is it then causes them to exert a force on each other. And this is the motor effect. When the magnetic field between the north and south poles interacts between the magnetic field of a current carrying wire, a force is then experienced. Now we can find out the direction of the force by using Fleming's left-hand rule. So we have the thumb. Now the thumb is the direction of the thumb is the motion. The first finger is the field. And the second finger is the current. And then you have your hand like that. So normally it would not be drawn like this. So it would be drawn, you have your north pole magnet here, you have your south pole magnet here, you have your wire, and therefore you have your magnetic field, and you have your even more magnetic field here, here, here. So we can use right hand's Fleming rule in this scenario. So we're told that the direction of the field is going from here to here. So you're going to put your finger that direction. And then we know that the current is going, let's say we're told current is going up. And then the field is going like that. And you realize that it's then the force is going to be moving that way. So the fullest force is actually at right angles. So if you wanted to maximize the force, then you would want it to put it at 90 degrees. There's also an equation which you recognize as F equals BIL. Force is equal to the magnetic flux density, which is represented by the B. So magnetic flux density multiplied by the current and multiply by the length of the conductor. Now we've got the motor effect down to a T. Let's look at, and we've got 11 maths done. We're going to now go into more complex ideas, but if you're able to understand this, then you'll definitely be fine and you shouldn't have to worry about the split ring commutator. So let's look at the split ring commutator. So a DC basic motor looks like this, first of all. So imagine that's the South Pole, that's the North Pole. We then have a dotted line here just to show. We've got the wire here, the wire here, and all controlled by this cylinder thing. And we've got positive on this side, and we've got negative on this side. So what does this mean, and what is this? So this is a DC motor, is a basic motor. So what happens is it maximizes the motor effect that we just discussed, in which the direction of the, let me do it in this. So the direction of the field is going from this way to this way. The direction, so your first finger will be going from there to there. And the direction of the current, which will be moved by the finger, is going there to there. So if you've got your first finger this way, your current that way, and therefore you will be experiencing a downward force. However, on this side, it will be totally different because it will be experiencing an opposite force. So what happens is one force goes up and one force goes down, and that's how the motor works. And what happens is this thing is called the split ring commutator. And the purpose of the split ring commutator is to change the contacts, which means change the positive and negative so that it keeps turning. Because otherwise, once it reaches the 90-degree angle of this going 90 degrees that way and that going 90 degrees this way, it will just stop moving. And if you want it to keep turning, you either have to swap the contacts or swap the pole magnets. So the split ring commutator is this key component here, and it swaps the components every half turn to keep the motor rotating in the same direction. Now, Fleming's left-hand rule can be used to deduce which way it will turn. So we're using Fleming's left-hand rule to see what way it can turn. Because the wire this side is going this way and the wire this side is going that way. Okay. We're now going to look at loudspeakers, headphones, or microphones, the generator effect, generators, microphones, and transformers. So, loudspeakers. Okay. So, loudspeakers, let's draw the diagram of loudspeakers. So, we have, as usual, the iron core. So, the iron core in the middle. And then we have the wire that's surrounding it. So, the coil of wire. Imagine if that was around it. Okay. And then this is connected to a paper cone. Let's do this in blue. And then we then have the north and south magnets above and below this. So we see the motor effect here as the magnetic field of the north and south is interacting with one another. So what happens here? Oh, so sorry, no. Actually, I just tricked myself. So what actually happens is this is the south magnet, and then this is another north magnet. So you can see that these two have an interacting magnetic field going from here to here. Then these two have an interacting magnetic field going from there to there. And then this also has its own magnetic field going from there to there. So we have three magnetic fields interacting in a loudspeaker. So let's imagine. So we begin with alternating current being sent through the coil. Oh, there's also just connections from here, like this. So we see alternating current being sent through the coil to the base of the paper cone. So this current is being sent to the base of the paper cone. This current is being sent. The coil surrounds one pole of a magnet and is surrounded by the other, causing a force on the coil. So we see a force being exerted here. Now, variations in the current makes the cone therefore vibrate at different amounts. So when we change the amount of current, we then change the force on the coil, and we then change the vibrations of the paper cone. And this is how different sounds are generated. Now we also have microphones. Now microphones are loudspeakers in reverse. If you can just imagine that. So what happens is these sound waves hit the diaphragm. This is then attached to a coil of wire wrapped around a magnet. The wire then moves into the magnetic field because it's just been hit. So it's pushed into the magnetic field. And the distance that's pushed creates a different effect in a different magnetic field and current, and therefore there's variations in the how far it's being hit, creates variations in the sound. So typically the frequency of the sound wave is the frequency of the alternating current. Now let's look at the generator effect. So the generator effect is the induction of a potential difference as well as a current, if there's a full circuit, in a wire which is moving relative to a magnetic field or experiencing a changing magnetic field. So the generator effect, in just simple terms, is when a potential difference is created or induced due to a changing magnetic field. So by moving the magnetic field lines in a coil of wire, or by moving a conductor in and out of the magnetic wire. Or you can also turn the magnet, which changes the magnetic field and induces a potential difference. Every half turn, it will reverse it. So changing the magnetic field can induce a current in a wire, created by the second magnetic field around that wire. The second magnetic field always acts against the true change that's made to return things back to where it was. So if you keep the magnet moving backwards and forwards, it then produces a potential difference that keeps swapping direction. Therefore, you produce an alternating current if it's a full circuit. Now, generators make use of the generator effect. So alternators generate alternating current. So whereas dynamos generate direct current. And they use different things. So the alternators have a similar construction to a motor. So let's just copy and paste this diagram and change what needs to be changed. So the alternators have construction just like a motor, but instead what we have is you also have a further one of this. So let's add the changes in red. We have another split ring commutator thing, which is actually called, this is a split ring commutator. This is called the slip ring and brushes. So instead of a split ring commutator, AC generators have these things called slip rings and brushes. And what they do is they have this big thing here. So this is a slip ring and this is a brush. And what they do is that they don't swap the contacts every half term. Every half turn, because we want the contacts to swap so that we can produce an alternating potential difference and an alternating current. Now dynamos are still a bit similar, so let's just duplicate this. However, we don't have this. We don't have slip rings and brushes. We have something else now. So you have split ring commutators for motors, for basic DC motors, direct current. For dynamos, we have something called a split ring commutator, which we've already looked at, as you know. Now, a split ring commutator is used in a basic dynamo, which we already looked at, which is the same way as alternators and just swaps the direction every half term to keep the direct current. So we were to look at the traces of them, the various traces. Traces are just like the graphs that each produce. So the alternating current trace would look like this. The direct current trace would look like just this. So it's still changing because the electrons are still also increasing revolutions. It's totally different. So now onto the final part of this topic, transformers. Before we do the small revision on space. So transformers change the potential difference of alternating current. There's step up and step down transformers. So this has quite a simple diagram, actually. Which is really hard to see, sorry. And then we have it returning back. Sorry about the internet issues if you are watching this. Okay. Okay, I'm unable to get my pen back, but okay. So then we have the rest of the circuit. So here is what we call one of them is going to be the primary coil, and one of them is going to be the secondary coil. Now one steps up the voltage and one steps down the voltage. And the magnetic field produced from both of these, the magnetic field produced from this coil to this coil looks like this. So the step-up transformer increases the potential difference. How does it increase potential difference? It does this by having more coils on the secondary coil than the primary coil. So step-up transformers have more coils on the secondary coil than on the primary coil. Now step-down transformers, you can imagine what this is, and it has more coils on the primary than the secondary coil, whereas this has more coils on the secondary than the primary. Now what happens is the alternating potential difference is therefore applied to the primary coil 1. So imagine the alternating potential difference is applied to primary coil 1. The iron core, because remember this is an iron core, then it magnetizes and demagnetizes really quickly, which induces a potential difference in the secondary coil. So the primary coil has a potential difference applied to it. It then magnetizes the secondary coil, quickly induces and demagnetizes it, the potential difference in the secondary coil. And if the secondary coil is in a complete circuit, then current is induced as well. Now the ratio goes like this. The voltage of the primary coil over the voltage of the secondary coil will be a ratio of the number of primary over the number of secondary, and vice versa. So if you want the voltage to step up, then the secondary coil has got to have more than the primary, and if you want it to step down, the primary coil has got to have more than the secondary. And that is the topic of electromagnetism, and now we're going to just cover the final topic of space. We're just going to spend this last couple of minutes going over the life cycle of stars and the solar system and orbits, hopefully, if we have time. So let's go over the life cycle of stars. You know what? Let's make this fancy. Okay. So let's go over the life cycle of stars. Okay. So the first step is that stars just initially form because of dust and gas. So there are dust and gas present in the universe, and they then fuse together to create what we call a nebula. Okay. Then the forces of gravity then pull nebula together and form a protostar, forming a so nebula are like the clouds of dust and gas in the sky, and the protostar is the forces of gravity pulling this nebula together. Okay. We then after the force of gravity pulls it together, the temperature then rises. And because the temperature rises, the star gets more dense as more particles collide. And as more particles collide with each other, the temperature eventually gets high enough for the hydrogen nuclei to fuse together to undergo nuclear fusion when two light particles join together to form one heavier particle, which in this case will form a helium nuclei. So you've got 1, 2, 3, 4. So step 1, there's dust and gas present in the nuclear in the galaxy, which is what we call nebula. Then the forces of gravity then pull the nebula together and create a protostar. The temperature then rises, increasing the density. The more particles collide, increasing the pressure. Then the temperature eventually gets high enough for nuclear fusion to take place, and the hydrogen then fuses together to form a helium nuclei. This then gives out a huge amount of energy. Now, this outward energy balances the inward energy, the inward collapsing force. Because there are forces causing the protostar to try and collapse inwards. Therefore, they are in a stable state now, in which there is a balance, and this long stable period happens over several billions of years. And is called the main sequence star, the middle of a star's life. Okay. So after the main sequence stage, which is after millions and millions of years, we then reach the fact that the hydrogen eventually runs out. And since the hydrogen runs out, the star then begins to fuse heavier elements. But it can only fuse heavier elements up to iron before it begins to swell and go red. This then forms a red giant, and even a red supergiant, depending on if it's a large star. Now, if it becomes red, it's because the surface is cooled. Fusion of other elements occur, and heavier elements up to iron are created in the core of the star. Then, from this point onwards, we have choices. So for step number 9, we then have either a red giant, and if it's a red giant, it then just becomes unstable. The red giant then becomes unstable. It ejects out a layer of dust and gas. It ejects out dust and gas and becomes a white dwarf. This is one similar to our sun. This white dwarf then fades into a black dwarf. However, if we have a larger star, then it's called a red supergiant. And this red supergiant then tends to glow quite brightly. It undergoes fusion. It expands until it undergoes a super, which is a massive explosion. All of these elements are then exploded and ejected into the universe to form new planets and new stars. And then the exploding supernova throws out the layers of dust and gas into space, so leaving a very dense core, which is known as a neutron star. And if the star is still big enough, then it can become a black hole, which is so big that it just light can't even escape from it. And that's the life cycle of stars. Well done for watching. If you got up to all 4 hours, I'm very proud of you for going this far. And yeah, it's almost the end of the first week of half term. So tell me how you're doing, and see you for the 9 o'clock one if you're going to come for English literature, English language, and GCSE.